So I actually have forgotten the codes, but hemoglobin beta and Homo sapiens should give good enough results. So the first two things are quality. I like UniProt because here you have one of the first one, the reviewed sequences versus the unreviewed sequences. So they are always going to be better when they are reviewed versus unreviewed. Then within pretty much every single entry, you find these two. Again, reviewed, annotation score. You want, well, these are not stars, but you want 5 circles. It doesn't sound as cool, right? 5 circles, but that's what you want in an annotation, 5 circles. Sometimes you have to do with 3, and ideally, if you have the option not to use something with less than 3, do it. But if you have no other option, well, you have no other option. Let's see, what else was there? Okay, pathology and biotech. So, anemias. A good enough answer for any hemoglobin subunit would be that is related to anemia. Why? Because that will be the most general disease or pathology associated with them. And they will have many forms. But lucky us, anemia is a very well understood noun. Of course, that's only true for hemoglobin. There's, of course, you were tasked to mention the difference between a natural variant, which are kind of like mutations, and over here in sequences, isoforms. Here in the instance of isoforms, these other sequences are of lower quality, and they are not really identified as the normal eukaryotic isoforms. And for this, if you haven't taken molecular biology, you wouldn't know that. But if you have isoforms in eukaryotes, mean alternative splicing or post-processing after gene translation. There's many instances of that. Insulin is one case, but there's plenty more with very, very wide complexity. If you don't find the sequence over here or the sequence with the difference spelled out, these isoforms might be inconsequential. And later I'll show you one where the information is actually useful and it tells you far more than this. What else was there? Yes, the properties. Here, this box for the sequence, if you click here in this menu, you get access to all of the properties. So isoelectric point, molecular weight, and several others, right? So that was ridiculously easy. Well, it was easy if you knew where to look. And we have been talking about that for several classes. There's many more tools, but those were the important ones for this test. We are going to talk about others, but from different databases. Is there any specific questions that you would like me to address from the UniProt here, now? You sure? Okay. We didn't use the basket. We didn't. Oh yes, we did some single sequence, pairwise alignments to identify the identity. Those were easy. If you use the basket, pretty much doing whatever you want with sequences is pretty straightforward. Now, both, actually not both. You'll notice that I didn't make any question for OMIM. It was only UniProt. The reason is going to become clearer a little bit later, but that is one of the difference, or the main difference with UniProt and OMIM. In OMIM, we are always restricted to humans, and there's no really a specific measure of the quality of the data. However, there is a semi-quantitative descriptor in the symbols. When we know the gene, when we know the molecular basis of the disease, and several other things. This one is harder to quantify, but sometimes can be done just by looking at the content of the respective site. For better or worse, and I'm gonna click on the first one, so if you wanna follow, please catch up, go to OMIM 141900. So these two, let me, okay, well, are you up to this point? No? Who's behind? If you need to catch up, let me know. We ready? Okay. So where I want to go, let me just check the gene structure. Okay, yeah, so this is words. What I want to start using is this, the cytogenetic location as well as the genomic coordinates. Both, or actually UniProt deals only with protein sequences. 20 different letters and that's it. OMIM deals mostly with the effects mutations of these proteins or natural variants have over the human body, and mostly pathologies. But we haven't talked about any or even just one DNA sequence, right? So there must be sequences for DNA. The reason I leave this one for last is because it's actually, at least to my taste, very difficult to work with. I already mentioned that the cytogenetic location refers to, by the first number, the chromosome, the letter P, either the long or the short arms of the chromosome, and 15.4 refers to a specific band. In cellular biology, we don't talk about this, but way back when, the chromosomes used to be stained with antibodies, and the number of bands was used to be counted to identify locations. You're probably more familiar with what is described over here. These are the genomic coordinates, and starts with the 11, again, the 11 chromosome, and then it has two numbers separated by a dash. This number, 5,225,465, is the number of the first nucleotide in the sequence, in the genomic sequence for hemoglobin beta. And it stretches, well, if you want to make the subtraction, it's about 2,000 nucleotides. I'm going to go and click there because that is going to take us to the next database we're going to talk about. And this one, this is a specific version of that database. This is part of the NCBI. Does it look easy to read? No? Well, it's not. It's actually quite complicated. So let's start from the simple parts. Up here, we actually have a very simple schematic view of the chromosome. We have chromosome 11, and here we have one of the arms, the one that corresponds to P, and on the other side, we have the one that corresponds to Q. Again, maybe in some cytology, I don't know if that class still exists in any program, but if you take an experimental approach to cell biology, you will look at what these bands of colors correspond. For the time being, doesn't matter. And here in this red line is the location of the sequence that we are looking at, the location of hemoglobin beta. So that one is fairly easy to read. Here we have the beginning, well, actually the range that we're looking at, 1,606 base pairs. When we're talking about DNA, it's assumed that it's always double-stranded, so the sequence is referred as base pairs. But because it's double-stranded, that means that if you have one strand, you still have that number of nucleotides. Now, who has a calculator in hand? Who's fast enough to divide this number, 1,666, by 3? The number of nucleotides that get translated to one amino acid. What's that?
Like
535 amino acids, right? Let's omit the point because you don't have half an amino acid. And remember the size of hemoglobin from the protein, from the UniProt? 150, let's say. So we have almost 5 times more information than actual amino acids. You know why that is, right? Why? because eukaryotes have introns. So not all of this sequence is going to be translated. And that is the reason why there's more isoforms for many other genes, because sometimes some exons are not used unless there's a specific mechanism and signal to do so. So we're always going to have more nucleotides than amino acids. Okay, yeah?
So the elements like enhancers and promoters and all those...
I usually not included here. Here, and this is a rule of thumb, that is, you need to check the database for what it's showing you. It should only include the open reading frames. That is everything and anything that is translated into protein. Any regulatory sequence should be either before or, well, again, I'm generalizing, before or after. Of course, there's genes with regulatory sequences in the middle, but I don't think hemoglobin does that. Okay, then we have this red line, which is the region we're looking at. And then, by definition, everything in this rectangle, it's assuming into that little tiny line. And here is the scale. As in any map, we need to know how big things are. This line, this black line, represents 500 bases. So we need about 3 of those to cover the whole gene. Here we have, let me remember. Oh yes, we have usually thin lines for genetic material that is not translated into protein. So we will have these thick lines representing the exons that actually get translated. Here we have regions of the protein, or so-called alleles here, where known changes are represented in the omic. Over here, what do we have here? Oh yeah. Yes, sorry. I usually lose track by now. Over here, we have the information that a program or the automated curation that tries to identify regulatory elements. Here in gray, DNase clusters. Do you know what a DNase is? No. DNase.
Enzyme?
It's an enzyme that degrades DNA. Now, also, way back when, like in the 80s, the only way to map the genome was not sequencing, but destroying DNA with DNase and map the regions that were not destroyed. Have you seen, who has not taken a molecular biology class? You are taking it? Have you seen what a nucleosome is? No? Who can explain what a nucleosome is? Nobody? So in the cell, DNA is not free. It's wound around histones. And the DNA that is wound around histones, it's not accessible for DNases. So it was an easy way to map what regions of the genomes were wound around the histones. Here is a very large, well, this is the result of an alignment, and it shows the conservation of this sequence with 100 vertebrates. Lines that are blue means very conserved. Lines that are red are very low conservation. And many, many other things. You know what a rhesus monkey is? Okay, well, here is the conservation with that. There is the conservation with mouse, dog, elephant, chicken, Genopus tropicalis, zebrafish, or Danio lamprey. And you can see that depending on the being, the animal, depending on the genome, there's very different degrees of conservation. So the purpose of this window is to give you a very quick overview to what is known for the genetic sequence. Not for the protein, but for the genes. Yes?
It's just that I moved the whole thing, and now, I don't know. I clicked on the rhesus, mouse, dog, chicken.
In that section?
And then I moved it, and now it won't move back.
Well, there's always reload and the back button. So look, if you look at the screen, there's actually many ways to navigate. For example, up here, you have these arrows. The tiny ones mean small movement. The one with 2 means a larger, and the ones with 3 means a very large movement. So let's make this exercise. Now that Emily is doing disasters in her computer, press the tiny arrow back. Oh, you sure? We move away from that gene, but not so much that we have other genes visible. It was a tiny move, right?
Yeah.
I'm going to undo it by going back. Oops. Okay, this is the view we started with, and I'm going to click on the one that is a very large move backwards. Now look at the numbers up here. 5,223,940, where we should be at 225. So we move a decent amount of nucleotides, but because the human genome is so big, there's actually nothing that much interesting back here. Just because of the location of the hemoglobin beta. That doesn't mean that the whole genome is going to be like that. There's going to be regions where we're going to have genes in both DNA chains, for example. And that's going to be hard to look at. I'm going to go back to where we started. There's other ways to navigate. We have zoom. We can get closer to the sequence 1.5 times, 3 times, 10 times, or even at the base level. So click on the base level. And look, now we can actually read the sequences for the human, for the rhesus monkey, for Genopus tropicalis, and if there was an alignment for the dog, the mouse, we could read the sequences. But just consider how many base pairs are there in the human genome. You remember that? You should know that. How many? No. Come on, be bold. About 6 billion, or that is 6,000 million base pairs. So we can, maybe we want that, but it would be very hard to look at all of those base pairs at the nucleotide level. So that's why this database starts with the zoom out that we had. Which actually I don't know how to go back. What do you think? 10 times or 100 times?
100.
100? Let's click on 100 then. Well, actually that was a bit much. Here we can see the regions of the hemoglobin beta, and we zoom out enough that we can see, look over there, hemoglobin delta. It's nearby, but according to this scale, it's actually about 5,000 base pairs away from hemoglobin beta. Okay, again, molecular biology. Over there should be downstream, over there should be upstream. 5' to 3'. Okay. Notice in this representation of the database, there's no information for the regulatory sequences, or at least none that we can see. There should be in another region of the database. Okay. Any questions thus far? You can see that the philosophy is totally different in this database. You do not only need to know a lot more about what you're reading, but the view is totally different, completely, absolutely different. For example, if we wanted to independently compare these sequences, there's no easy way for us to retrieve them, or just even to download this one. And this is something that I didn't show in the slides, but I should have. There's two very quite different databases in terms of who can read them. UniProt is easy to read for a human, right? Well, fairly easy to read for a human. This one, it's almost impossible to read for a human, and it's more easy to read through a computer or by a computer. And I'm talking about programming so that you don't have to parse this information and a program does it for you, which is pretty much this viewer. This is probably a Java applet or something like that that is transforming a text into this graphical information. That's one reason I don't really like it that much, because if I want to read about one gene, I really have to understand tons of things that may not be relevant for what I want to know. If you want to read about thousands of genes, probably this is way better. Any questions over here? You sure? Good. It's important to be clear about these things.
Just at the, I mean, adults? No, like.
Here.
Just, what is that exactly?
So these should be variations. Let's see. Probably to understand this better, we should go back here. But why don't you try click on this? Sometimes databases have the help hidden. And the best way to find the help is click on the words. Did it do something? No? Okay, well, that was not the case. It only highlights where you are pointing. So let's go back to the OMIM database and let's see. Let's go to this section of the text that is the allelic variants. Okay, so I click on allelic variants and it moved me in the OMIM to these variations of hemoglobin. Here, for example, the Alborg has a mutation of a glycine for an arginine. Who can tell me, using whatever information you have in hand, how different is glycine for arginine, from arginine? Just the charges?
No, the size.
The size is a huge difference. This one has a hydrogen for a side chain, and this one has it's not an urea. Well, that's something that looks like urea and other carbons. It's like having an arm and then only having up to your shoulder. It's a dramatic difference. It's the charge, but much more. So this mutation is an allelic variant. There is another one, histidine 143 for arginine. Does that number sound familiar? Is that the histidine that binds the iron? If it's so, this mutation could be very bad because arginine is way bigger than a histidine. Anyway, those are the allelic variants. There's this view that I'm in, and there's also the table view, which is usually more useful because you can get a look at all of them quickly, and you get to see the mutations. If you have a table, what's that? It's not seen anymore. Table. What happened to my bar?
¿Cómo podemos ver los allelic variants?
So first, allelic variants, and then table view.
Allelic variants son mutaciones o polimorfismos o qué son.
That's a good question. It will depend on the frequency. Again, if it's only 1%, it will be a mutation, right? Above 1%.
No.
I'm saying the other way around.
Yeah. 1% is
Less than 1%.
Is a mutation.
It's a mutation. More than 1%
polymorphism.
will be a polymorphism. I don't know enough about any of this to tell you all of them are, none of them are, or some. But that will depend again on the details of how many times it's manifested, right? But these are known as variants. At the worst case scenario, you could describe them all as mutations. They are different sequences.
There is people that say that allelic variant is the same that appears.
I don't remember the definition of allelic variant including polymorphism and excluding mutations. And the only reason I say so is this. If you don't know, if you haven't sequenced it, and it's a dominant variation in a population, you cannot consider it an allele, right? Not quite. What is your source? Wikipedia?
No.
Nah, just kidding. Go ahead and check. However, this table contains the mutations, the name they were identified, and the clinical relevance of these mutations. Okay, so did that answer these OMIM alleles? Okay. So we got here to the NCBI through the OMIM. This is not the only way to do so. As you can see on these external links, we also have genome DNA links to other databases where if we want just the sequence for this one might be easier. For example, assemble the genome viewer, that's where we were. Sorry, not the NCBI genome viewer, but the UCSC genome browser. Assemble for the genome or just the DNA. And assemble NCBI RefSeg UCSF genome browser. So this time, let's go straight to the NCBI on the DNA section, and let's see if we can actually recover the information. And this is what I was talking about. First, Homo sapiens hemoglobin beta mRNA. So that means this sequence first is RNA, and second, it does not include the introns. This is only going to be the exons. So if we were to quantify how many base pairs, it should be more closely to what the the residues say. So get some guys. So 628 divided by 3. Come on, come on.
9. Son 9. 27.
209.
209, and over here we had 147. So there's still a discrepancy, but we are closer to what we are supposed to have. What could be the difference? Well, what happens to mRNA in eukaryotes? It gets polyadenylated. It also gets capped with a guanosine, methyl guanosine or something like that. There's modifications to mRNA, and those could be included in the sequence there. So I'm just trying to highlight some of the differences of going from protein sequences to genomic to mRNA. You always have to be aware of that, because if you are not, sometimes you can have sequences that seemingly don't make sense, but it's because the origin of the sequence. What, is this ugly or beautiful? Easy to read, hard to read? What do you think? I have no idea what's going on, right? Also, it's trying to be kind of accessible, and it has some words in capital letters. These are the equivalents to these nice and highlighted menus. For better or worse, you cannot click on any of these, but you can read them. And at least we know that the definition of this sequence is Homo sapiens hemoglobin subunit beta. And this is an mRNA. It's from Homo sapiens. We have again all of the phylogeny and different papers that relate to the sequences. Because this is hemoglobin, and everybody works in hemoglobin, and I guess every MD in the world works with hemoglobin. If you scroll down, it's going to be forever and ever that we're going to see references, right? It's going to be several. Why everybody works with this? Can we easily find a rating for the quality of the data? Not really. At best, we have this comment. This record has been curated by the NCBI staff. The reference sequence was derived from this and this. This sequence is a reference standard in the Genome Project. On June 13, 2018, this sequence was replaced by another. So this only adds to the confusion, and this is something that I don't like about this database, that if you are not careful, you are looking at an old version. And you need to go through these comments to find out which is the most recent. And then, We still have to scroll down. Here is the protein sequence. Go down, and over here we finally have the mRNA sequence. Is it easy even just to click, select, and paste elsewhere? No. I don't know why they have made this database like this, but this is the way it is. However, if you are crafty enough, you can select that, paste it on Word or other editors, remove the spaces, remove the numbers, and then you have the sequence. Now, this is mRNA, so it's going to be directly translated to protein, right? This already should contain the codons to start the protein translation and to finish it. So we should be able to find, do you have your tables of codons? What is the start codon? AUG? Where is it? Notice that for whatever reason this database doesn't change the T's for use. So what we should be looking is for ATG. Well, there's one over here, but it's way ahead, right? We don't know if it's the right one. And because of this type of database, while the information should be there, we don't know where it is. We cannot find it easily. But wait, this is the things. This is the way things are in this database.
The first line.
What's that?
ATG.
ATG?
On the first line.
Yeah. Is it the first one? Yeah. So this is what? 10, 20, 30, 40, 50 nucleotides in, right? But I might be wrong. We need to go through this information to find out if what I'm saying makes sense. Now here, confusingly enough, at the top of the page, you have these links. You can click, don't click on FASTA, click on graphics. Now we have a view similar to the browser we have earlier. Just by clicking on graphics, now we can see the gene HBB. Don't read it on the screen, try to read it in your computers. The name of the reference sequence and a host of information, including the exons of the gene. This is totally a different view from the genome one, where we had all the introns, all of the exons, and more sequence. Here is restricted only to this sequence, the mRNA. And if you click on FASTA, you should get exclusively the sequence. FASTA. So I'm going to go back to the graphic view. You have GenBank, which is the view we started with, the hardest to read. You can click on graphics, it will send you here. And if you click on FASTA, it will send you only to the sequence. I generally hope you don't have to work with this because it's really confusing at times. It's not really fast to use if you are doing it on your own. But you can find the information, at least. For better or worse, we still do have tools as BLAST. But this BLAST is going to look for nucleotide sequences that are similar to this one. It's not going to look for protein sequences, nucleotides. So if you find a sequence that looks like that, it will only produce a protein like hemoglobin if the start codon or the end codon corresponds roughly to a protein of the same size. Which is to say, you can have pseudogenes that lack the start or the end and have exactly the same sequence anywhere in the genome. But because they don't have a translation code, they are never going to be genes. So be careful when you do BLAST for genomic sequences, even for mRNA sequences. They could be nothing, ghost, or DNA junk, if you will. Okay, this is the worst database in my opinion, but hey, I cannot skip teaching this. You might need it. I'm going back to the graphical version.
So this is
Yes. Yes. For better or worse, at least that's included on the title. This is not the genome. The genome was in the other view. This is the mRNA. So, you could try that. For example, a very simple exercise that could be your homework is get the genomic sequence of hemoglobin beta, get the sequence from the mRNA, and align them. That alignment should tell you where the exons were and where are the introns, right? Shh, don't tell them. If you can do the homework easily, just do it. Or charge for the answer.
And where do we align it?
Aha, that's a very good question. Well, technically you could do it by hand, right? What do you think 500 bases is a lot? Yeah?
Which sequences?
The genomic sequence for hemoglobin beta and the sequence for hemoglobin beta but from the mRNA. This is something that will help you. Remember that you could get to UniProt using UniProt, but there is an XPaSI version of UniProt. So if you go to XPaSI, over here to the left, we have resources. And these resources should include genomics and, well, now I guess it's under genomics. And there you should be able to find sequence alignment. So there you could align, well, actually, that one is very limited. Let's see similarity search. Over there, of course. Over here you find plenty of tools to do sequence alignment. If I may recommend one, I will go for T-Coffee.
T-coffee?
T-Coffee. Almost at the bottom. But wait, before you click on T-Coffee, whenever you need a tool for molecular biology, proteomics, whatever, in that regard, bioinformatics, always come to XPaSI first. This database is actually full of tools that are very useful, and because you can just click at every one of them, you can take the same information, try different programs, and decide what suits you better. I'm not going to cover them all because, as you can see, just for sequence alignment, look at the diversity. Okay, as I said, go for T-Coffee. And let's see, this link is, so here you have it, alignment, T-Coffee, DNA, RNA, or proteins. I will start with T-Coffee itself, not with M or R. Go for T-Coffee, get the sequence from the genomic sequence for hemoglobin, get the sequence from the mRNA, align them. And ideally, the gaps between one and the other should be the introns.
Pero ¿de dónde sacamos el DNA?
El problema es que no entra.
In the genomic view,
Pero es T-coffee.
yeah, you need the FASTA from the mRNA and the FASTA from the genomic view.
Okay, we have to copy the sequence here.
Yeah. This time we don't have a basket, so it's not as easy. You need to copy the sequence to a text editor. Well, copy both sequences to a text editor, then go to T-Coffee, which is going to have a window like a text editor, and you paste at them and click submit. What would be the difference? Now, usually, look at, it's a good question. Here, paste or upload your set of sequences in FASTA format. Sequences to align. Can you read what it says here? Click here to use sample file. Well, click there. And look what it does. It's using proteins, and it has first one protein, then the next, and several. 1, 2, 3, 4, 5. 5 sequences, one after the other.
Entonces sí.
What?
Entonces, sí,
Yeah.
pegamos.
One after the other. First the genomic, then the mRNA, and click submit.
Do we use the sequence from NCBI?
Yeah.
It's easier.
Yeah. Wherever they are deposited, they should be pretty much the same. But you need to use the mRNA and the genomic one.
But you said we need to delete the numbers and the spaces if we use.
Yeah, so reduce them to something like this.
And
Oh, you copy with the number for nucleotides?
yes.
Yeah, well, of course. Look at this. This has no numbers, no spaces.
Understand?
Yeah, that's one of the rules. No numbers, no spaces. Anyway, that's a homework, so work on that. Any other questions? Well, make an effort. You have only spent like 30 seconds. Any questions? No? Okay, well,